After fertilization in angiosperms or flowering plants, whatever the changes can take place in a flowering plants or in angiosperms after fertilization is called as post-fertilization events. In this class, we discuss post-fertilization structures and events. Generally, before going to that, post-fertilization structures and events, first we discuss fertilization. Generally, this is the pollen tube. The entry of pollen tube into embryo sac is attracted by filiform apparatus present in synergids. So that's why the growth of pollen tube in angiosperms is chemotactic and apical. That means from the apical part of micropyle. The growth of pollen tube in flowering plants during fertilization is chemotactic and apical. And whenever the pollen tube enters into embryo sac through the degenerated synergid, it is attracted by filiform apparatus. At the tip of this pollen tube, a pore-like structure is formed. Then two male gametes are discharges into embryo sac. The first male gamete move towards egg cell. The male gamete haploid and egg cell haploid, these two are physically contact to each other by the process of fertilization or syngamy. Then it's formed a diploid zygote, and the diploid zygote can develop into a diploid embryo. And the second male gamete releases into embryo sac. It moves towards center of embryo sac. The center of embryo sac having a largest cell, that is central cell. During fertilization, the two haploid polar nuclei fuses to form secondary nucleus. The ploid of secondary nucleus is diploid. Now the diploid secondary nucleus having two nucleus and one haploid male nucleus is physically contact to each other by the process of triple fusion to form a triploid primary endosperm nucleus or pen. So, in embryo sac or female gametophyte, two different types of fertilization can take place. One is syngamy, another one is triple fusion. Both syngamy and triple fusion is called as double fertilization. In double fertilization, syngamy is called true fertilization and triple fusion is called vegetative fertilization. The product of syngamy is a diploid embryo. The product of triple fusion is triploid primary endosperm nucleus. Then double fertilization can take place. So presence of double fertilization is an important characteristic features of angiosperms or flowering plants. So whenever fertilization is happen, after fertilization, a changes can take place in flowering plants is called as post-fertilization events or structures. In flowering plants or in angiosperms, there are four different types of post-fertilization events can take place. Such as the first post-fertilization event is endosperm development. The second post-fertilization structures and events is embryo development. The third post-fertilization event is fruit formation, and the fourth post-fertilization event is seed formation. These are the four different post-fertilization events can take place in angiosperms. Let's we discuss the first and very most important post-fertilization event is endosperm development. How endosperm development can take place in angiosperms or in flowering plants? Generally, the first and very most important characteristic features of endosperm development. Endosperm is formed by the process of triple fusion. So the first very most important point, endosperm is the product of triple fusion. When the second haploid male gamete fuses with diploid secondary nucleus to form triploid primary endosperm nucleus, these triploid primary endosperm nucleus can develop into triploid endosperm. So the development of endosperm or formation of endosperm is the product of triple fusion. It is the first characteristic features of endosperm. The second characteristic features of endosperm is, so endosperm is formed. Endosperm is formed from central cell of embryo sac. The largest cell in embryo sac is central cell. The central cell having secondary nucleus. It is physically contact with haploid male gamete by the process of triple fusion to form primary endosperm nucleus. Primary endosperm nucleus can develop into endosperm. That means endosperm is the product of central cell of embryo sac. And the third characteristic features of endosperm is, generally the ploidy of endosperm is in angiosperms. In angiosperms, the ploidy of endosperm is triploid. But whereas in gymnosperms, ploidy of endosperm is haploid. Reason is, in angiosperms, endosperm is formed after fertilization. In gymnosperm, endosperm is formed before fertilization. So that's why the ploidy of endosperm in gymnosperms is haploid, in angiosperms is triploid. There is one plant, the name of plant is called Enothera lamarckiana. The plant of Enothera lamarckiana, it is also produces endosperm in angiosperms, but its ploidy is diploid. It is a special characteristic features of this plant. And one more, after triple fusion, the product of triple fusion is endosperm. This endosperm, it's rich of reserved food material or nutritive tissue. Endosperm is a rich of food or nutritive substances. Whenever the endosperm is rich of nutritive substances, it is nourishing to growing embryo. The sixth characteristic feature is endosperm is nourishing or providing nutrition to growing embryo. The next general characteristic features of endosperm. Generally, the nature of endosperm is not similar in all seeds. For example, in the plants of cereals, example rice plant and wheat plant. In the plant of cereals, rice and wheat, the nature of endosperm is cellulosic. That means starchy in nature. That means the endosperm in cereal plants like rice and wheat, it stores food in the form of starch. But whereas one more cereal plant also is there, that is maize. The nature of endosperm in the maize is not starchy, it is proteinaceous. That means it stores food in the form of proteins. But whereas in the plants of castor, the nature of endosperm is oily. Whereas in the plant of date palm, hemicellulosic. So these are the best example that storage of food material in endosperm is not constant. It varies from one species to another species. Generally, in some family members, the endosperm formation is stops or prevented. Example, in the family of Trapeziaceae and Podostomanaceae, and in the plant of Orchidaceae, in the family. In the family of Orchidaceae, in the family of Podostomanaceae, in the family of Trapeziaceae, endosperm formation stops. In which of the following families endosperm formation stops means are not producers? Trapeziaceae, Podostomanaceae, Orchidaceae. These are the general characteristic features of endosperm. Once again, I recall post-fertilization events. Generally, there are 4 post-fertilization events in flowering plants or angiosperms. The first one is endosperm development, second one is embryo development, third one is fruit formation, fourth one is seed formation. The first post-fertilization structure and event is endosperm development. The first characteristic feature, endosperm is the product of triple fusion during double fertilization. And endosperm is formed from the central cell of embryo sac. Second characteristic feature. Third one is the ploidy of endosperm in angiosperms is triploid. Region is in angiosperm, endosperm is formed after triple fusion. Whereas in gymnosperms, the ploidy of endosperm is haploid. Region, the endosperm is formed in gymnosperm before fertilization. In the plant of Winothera or Enothera lamarckiana, endosperm is formed, but its ploidy is diploid. Generally, the endosperm, it stores food material or nutritive. Whenever it stores food material and its help for supply the nutrition to growing embryo. That means endosperm nourishing the growing embryo. And the nature of this endosperm is not same in all plants. For example, in the plants of cereals like rice and wheat, the nature of this endosperm is starchy or cellulosic. But whereas in cereal plants like maize, the endosperm stores food in the form of proteinaceous. In the castor, the nature of endosperm is oily. In the plants of date palm, Phoenix dactylifera, the nature of endosperm is hemicellulosic. In this class, now we discuss what are the methods of development of endosperm. There are 2 different methods of endosperm formation in flowering plants. The first method is free nuclear endosperm formation. The second one is cellular endosperm formation. Free nuclear endosperm formation is the first type of endosperm development. Cellular endosperm formation is the second type of endosperm formation. We discuss now the development of endosperm in angiosperms by 2 different ways. The first way of endosperm formation in angiosperms is free nuclear endosperm formation. The second one is cellular endosperm formation. Out of these 2 ways of method of formation of endosperm in angiosperms, the most common type of endosperm formation is free nuclear endosperm formation. What is free nuclear endosperm formation? Very simple. For example, this is embryo sac or female gametophyte. After fertilization, the zygote develops into embryo and primary endosperm nucleus. Due to triple fusion, primary endosperm nucleus is formed. The primary endosperm nucleus, it's a triploid. Ploidy is triploid in nature. 3 sets of chromosome in each cell, that is triploid. The single nucleus of primary endosperm nucleus undergoes nuclear division or karyokinesis. Many nucleases forms. But each karyokinesis immediately not followed by any cytokinesis. If the karyokinesis is not followed by cytokinesis, then this endosperm is called as free nuclear endosperm. At the end of embryo sac or at the end of a last stage, when all the free nucleus is formed, at last stage each nucleus is surrounded by cell wall by the process of cytokinesis. But karyokinesis immediately not followed by cytokinesis. This is a most common type of endosperm formation in angiosperms. The next second method of formation of endosperm in angiosperms is cellular endosperm formation. Cellular endosperm formation means this is female gametophyte or embryo sac. This is embryo. The nourishing embryo or embryo getting nutrition from the endosperm, you know very well. When the triploid primary endosperm nucleus undergoes many nuclear division and it forms nucleus after every karyokinesis immediately followed by cytokinesis. Immediately followed by cytokinesis. After karyokinesis immediately followed by cytokinesis, this method of endosperm is called as cellular endosperm. These are the two different methods of formation of endosperm in angiosperms. For example, the tender coconut having two different types of endosperms. The coconut having how many endosperms? Two different types of endosperm. The free nuclear endosperm. The free nuclear endosperm is coconut water. It is surrounded by the free nuclear endosperm or coconut water. It is surrounded by white kernel. The white kernel is nothing but cellular endosperm. Cellular endosperm. These are the two different types of endosperms we are seen in tender coconut. Let's discuss what is the fate of endosperm. After formation of these endosperms in angiosperms or flowering plants, what is the fate of this endosperm? Generally, this is seed. Inside the seed, a diploid embryo is present. Inside the seed, diploid embryo is present. The growing seed, it is getting nutrition from the endosperm. The ploidy of endosperm in angiosperms is triploid. Now, what is the fate of this endosperm in the seeds? So, because of this characteristic feature based on the fate of endosperm in the seeds of angiosperms, endosperm is classified into two types. The seeds classified into two types. One is endospermic seeds is also called as albuminous seeds. The second type of seeds is X-albuminous seeds is also called as non-endospermic seeds. The best example for endospermic seeds or albuminous seeds is rice, wheat and barley and castor. The best example for non-endospermic or X-albuminous seeds is pea seeds and groundnut seeds and bean seeds. Sir, what is the fate of endosperm in the developing seeds? Generally, the growing embryo getting nourishment from the endosperm. Reason is endosperm is a nutritive tissue. It's result food material. At mature condition of this seed, before maturation of this embryo, if the endosperm is completely used, this is one seed. The seed having triploid nutritive tissue endosperm and a diploid embryo. The diploid embryo getting nourishment from the endosperm. But in the mature seeds, before maturation of embryo is not completely mature, but before maturation of embryo, if the endosperm is completely consumed, such kind of seeds are called as X-albuminous seeds or non-endospermic seeds. Example, pea seeds, groundnut seeds, bean seeds, all Fabaceae family member seeds. Whereas, this is one seed, embryo, endosperm. Embryo getting nourishment from the endosperm. After maturation of embryo in the seed, still endosperm is present in the seeds is called as endospermic seeds or albuminous seeds. Example, rice seeds and wheat seeds and barley seeds and castor seeds. All endospermic seeds are full of, monocotyledon seeds are full of endospermic seeds. Once again, I repeat the differences between endospermic seeds and non-endospermic seeds. Endospermic seeds is also called as albuminous seeds. Example, all monocotyledon seeds are endospermic seeds. Example, rice and wheat and barley and castor. X-albuminous seeds is also called as non-endospermic seeds. Example, all Fabaceae family member seeds, bean and peas and groundnuts. Sir, what is albuminous or endospermic seeds? Even after maturation of embryo, still endosperm is present in the seed is called endospermic seeds or albuminous seeds. Before maturation of embryo, if endosperm is completely consumed in the seeds is called as X-albuminous seeds or non-endospermic seeds. Example, pea seeds, groundnut seeds, bean seeds. This is one of the first post-fertilization event in a flowering plant, that is endosperm formation. Let's discuss the second important event is embryo development. The embryo development, it is always precedes the formation of endosperm in the seeds. When the endosperm starts to form, then embryo starts to development. Before endosperm formation, embryo or not starts to develop. When endosperm is formed, then embryo starts to growth or development. So that's why the growing embryo getting nourishment from the endospermic tissue. In this class, we discuss embryogenesis or embryo development in dicotyledons and monocotyledons. Let's discuss. Out of 4 post-fertilization structures and events, the second post-fertilization event is embryo development, or simply we called as embryogenesis. Before going to the development of embryo, let's discuss what is embryogenesis. When an haploid male gamete contact with haploid female gamete by the process of fertilization or syngamy to form diploid zygote. Now this diploid zygote is a single cell or unicellular. The entire organism starts with single cell. The name of single cell is called as zygote, but its ploid is diploid. The development of diploid embryo from diploid zygote by require 2 events. One is cell division, another one is cell differentiation. Due to mitotic cell division and cell differentiation, the diploid zygote can develop into diploid embryo. This phenomenon is called embryogenesis. From zygote to embryo development, in between, what are the events can takes place, we discuss now. In this class, now we discuss the process of embryo development in dicotyledons or dicot plants. In dicotyledon plants, the process of embryo development, it is started with a single cell diploid is called as zygote. When the zygote is undergoes transverse division, it produces two unequal cells. The smaller cell is called as embryonal cell or terminal cell. It is present towards chalazal end or antipodal side of embryo sac. And the larger cell is called as suspensor cell. It is present towards micropyle end of ovule or megasporangium. When the zygote, it is undergoes mitotic division, it produces two unequal cells during embryo development in dicotyledon plants. Smaller cell is called embryonal cell, larger cell is called suspensor cell. Embryonal cell is also called terminal cell, is present towards chalazal end. Larger cell is called suspensor cell, is present towards micropyle end. Now, the second step of embryo development in dicotyledon plants are dicots. The embryonal cell is not divide anything now, it keeps same. Then the larger cell or suspensor cell starts to divide many transverse divisions, then it produces 6 to 10 celled suspensor. So, when the suspensor cell is divided transversely many times, it produces 6 to 10 celled suspensor. Out of these 6 to 10 celled suspensor, the first suspensor cell is large size. It is called as haustorial cell, is also called as vesicular cell. And the last cell in suspensor is called as hypophysis. Its main function is formation of radical tip or is nothing but root tip. The formation of root tip or radical tip in dicot embryo, because of hypophysis, it is the last cell of suspensor. Is it clear? Now, in the third step of embryo development in dicotyledons, the embryonal cell or terminal cell present towards chalazal end, it starts to divide. When the single embryonal cell vertically divides 2 times, transversely divide 1 time, then it produces 8-celled embryo. This 8-celled embryo is called as octant stage. Octa means 8. When the embryonal cell vertically divide 2 times, transversely 1 time, then it produces 8-celled embryo or octant stage embryo. In 8-celled embryo, outer 4 cells are called as outer tire, inner 4 cells are called as inner tire. The outer 4 cells of octant embryo or 8-celled embryo is called outer tire. It's help for formation of 2 cotyledons. Dicotyledon embryo having how many cotyledons? 2. And plumule. Example, these 2 cotyledons and plumule in dicot embryo formed by outer tire or outer 4 cells of octant embryo. Our inner 4 cells of octant embryo is called as inner tire. It is responsible for formation of radical and hypocotyle. Radical means root and hypocotyle. The radical and hypocotyle in dicot embryo formed by inner 4 cells of 8-celled embryo. Outer 4 cells of embryo forms 2 cotyledons and plumule. In the fourth step of embryo development in angiosperms or flowering plants. Now these octant stage of embryo divides periclinally, that means transversely and vertically is called as, both is called as periclinal division. Then it produces globular stage of embryo, multicellular globular stage of embryo. It looks like globe shape. Now this globular stage of embryo, outermost layer of cells forms protoderm, it forms epidermis in the root transfer section. And the second layers of these globular stage of embryo forms ground meristem, that means cortex and endodermis. The inner mass of this globular stage of embryo is called procambium. It forms vascular tissues like xylem and phloem. At final stage, this globular stage of embryo divides and differentiate to form heart-shaped embryo. And finally, the heart-shaped embryo differentiate to form mature embryo in dicotyledons. In the structure of mature embryo in dicotyledon plants or dicotyledons, it is having two cotyledons in the embryo. And it is having radicle. Radicle develops into root system. And it has a plumule. Plumule develops into shoot system. The dicotyledon mature embryo having two main parts. One is embryonal axis and another one is cotyledons. So the structure of dicotyledon embryo having two cotyledons, 1, 2. And the embryonal axis having plumule and hypocotyle and epicotyle. The embryonal axis in dicotyl embryo having plumule, epicotyle, hypocotyle. And the second structure is two cotyledons. The plumule can develop into shoot system, the radicle can develop into root system. The radicle surrounded by a cap-like structure is called as root cap for protection. And now the question is, these are the two cotyledons in mature embryo. Below the cotyledons in dicotyledon embryo, a cylindrical portion is called as hypocotyle. Above these two cotyledons, the portion is called as epicotyle. Plumule, epicotyle, hypocotyle, radicle, root cap, two cotyledons. These are the main labels involved in the structure of dicotyledon embryo. The process of embryogenesis in dicotyledons and the process of embryogenesis in monocotyledons more or less same. The next we discuss structure of monocotyledon embryo. Structure of embryo in monocotyledons. Monocots means a seeds of the plants having single cotyledon is called as monocotyledons. In this class we discuss structure of embryo in monocotyledons or monocots. Already I told you the process of formation of embryo development in dicotyledons and monocotyledons more or less same. After embryo development or embryogenesis, a mature embryo forms in monocotyledons. This is the basic structure of mature embryo in monocotyledons. In monocotyledon embryo, single cotyledon is present. That single cotyledon in monocot embryo is called as scutellum. The location of the single cotyledon in monocot embryo is lateral side. This is very most important characteristic feature. The location of single cotyledon in monocot embryo, that is scutellum, is lateral side. And one more important characteristic feature, the embryonal axis of monocotyledon embryo having plumule and radicle. The radicle develops into root system and the plumule develops into shoot system. In monocotyledons, one more very most important characteristic feature, the plumule produces shoot system, the plumule surrounded by a mass of undifferentiated sheath is called as coleoptile, and the root surrounded by root cap, the root tip surrounded by a mass of undifferentiated sheath is called as coleorhiza. Presence of coleorhiza and coleoptile is the characteristic features of monocotyledon embryo. These are absent in dicotyledon embryo. In grasses, the second cotyledon is start to develop. A second cotyledon, generally in monocotyledon embryo, single cotyledon is present, is called as scutellum. It is located at lateral side. But in grasses, these are also monocotyledon. Single cotyledon is present, scutellum, but second cotyledon is start to develop, but it not completely developed. Then undeveloped or underdeveloped second cotyledon in grasses is called as epiblast. It's very most important characteristic feature. In this class, we discuss the comparative study of monocotyledon embryo and dicotyledon embryo. We discuss now. Okay? Here, this is the structure of dicotyledon embryo. In the structure of dicotyledon embryo, this is the basic structure. In this basic structure, these two are called as cotyledons. The number of cotyledons in dicotyledon embryo is 2, whereas in monocotyledon embryo 1. The single cotyledon is called scutellum. And the embryonal axis having plumule develops into shoot system, and radical develops into root system. In dicotyledon embryo, the radical surrounded by a protective structure is called as root cap. And one more, in embryonal axis of dicotyledon embryo, above the two cotyledons is called as, the region is called as epicotyle, and below the cotyledons is called as, a cylindrical portion is called as hypocotyle. But root and shoot, radical and plumule not surrounded by coleorhiza and coleoptile, but we are seen in monocotyledon embryo. And very most simplest flowchart I am giving for you, synopsis for embryogenesis in dicotyledons and monocotyledons. Generally, zygote is the single cell. Whenever it is undergoes mitotic cell division, it produces two-celled embryo. For easy to understand, I am giving this flowchart. And now this two-celled embryo undergoes mitotic division, then it produces four-celled embryo. When the four-celled embryo undergoes mitotic division, then it produces eight-celled embryo, that is called arctant stage. Now this eight-celled embryo undergoes periclinal division, that means longitudinal division and transverse division, is called as globular stage of embryo. Then the globular stage of embryo differentiate to form heart-shaped embryo. Then heart-shaped embryo differentiate to form mature embryo. The embryo is diploid. The formation of diploid mature embryo from diploid zygote by mitotic cell division and cell differentiation is called as embryogenesis. The process of embryogenesis more or less same in dicotyledon embryo development and monocotyledon embryo development. These are the characteristic feature. The second post-fertilization structure and event is embryo development. Let's we discuss the third post-fertilization structure and event. Third post-fertilization structure and event is fruit formation. Generally, in the structure of bisexual flower, the innermost whorl is called gynoecium. The units of gynoecium is called carpels or pistil. Each carpel or pistil having three parts: stigma, style, ovary. Inside the ovary, ovules are present. Whenever fertilization can take place, simple definition for fruit is a fertilized ovary. A fertilized ovary is called as fruit. It is the simplest definition for fruit. When the ovary is fertilized, then fruits are produced. And inside the ovary, ovules are present. When ovules are fertilized, seeds are produced. This is the reason seeds are present inside the fruit. And this is ovary wall. During fertilization, the ovary wall can turn into fruit wall. Fruit wall is also called as pericarp. It's a protecting layer or protecting structure in fruits. Now the question is, this ovary can develop into fruit with fertilization or without fertilization. Only ovary can develop into fruit or other parts of the flower can develop into fruit. Because of this characteristic feature, fruits is of three types, such as first type of fruit is true fruits. Example, mango. Mangifera indica, discovered by Linnaeus scientist Dot. It's a national fruit. It's a true fruit. What is true fruit, sir? Only ovary can fertilize into fruit is called as true fruits. The second type of fruit is false fruits. False fruits means along with ovary, other parts of the flower, either pedicel or maybe thalamus. Either pedicel or thalamus along with ovary can develop into fruit is called as false fruits. During fertilization, along with ovary, other parts of the flower such as thalamus and pedicel can develop into fruit is called as false fruits. Example for false fruits is apple and strawberry and cashew nuts. These are the examples for false fruits. In these three different fruits, the edible part is thalamus. The edible part is thalamus. Reason is, in false fruits like apple and strawberry and cashew nut, along with ovary, thalamus can develop into fruit. So, in apple and strawberry and cashew nut, the edible part is thalamus. And the last and final most type of fruit is parthenocarpic fruits. Example, banana and seedless grapes. What is parthenocarpic fruits? Generally, ovary fertilized into fruit, but without fertilization, ovary can develop into fruits is called as parthenocarpic fruits. Example, banana and seedless grapes. The parthenocarpic fruits produced either by naturally or maybe artificially. Artificially, when we are using a growth regulator or growth hormone auxins, if you spray the auxins, growth regulator, the ovary without fertilization develops into fruit. Such kind of fruits are called as parthenocarpic fruits. When the ovary is not fertilized, then ovule also not fertilized, then those fruits are seedless fruits. A seedless fruits are produced without fertilization is called as parthenocarpic fruits. Our simplest definition, without fertilization, ovary can develop into fruit is called as parthenocarpic fruits. The process of formation of fruit in a flowering plants, there are three different types of stimulus responsible for formation of fruit in a flowering plants. The first stimulus is for formation of fruits in flowering plants by pollination. When pollination takes place, there is a first stimulus can takes place for formation of fruits in flowering plants. The second stimulus is seed start to formation. Seed start to formation. When the ovule converted into seed, when the seed start to develop or seed start to formation, there is a second stimulus for development and formation of fruit in flowering plants. And the third stimulus received by the plants for formation and development of fruits in flowering plants is nutrients. When macro and micro nutrients are available, then there is a growth and formation of fruits can takes place. These are the three different stimulus can takes place for formation of fruits in flowering plants. This is third post fertilization structure and event. The last and final most post fertilization event in flowering plants is seed formation. The simplest definition for seed is a fertilized ovule is called as seed. If you can imagine the structure of pistil or carpel, each pistil having three major parts: stigma, style, ovary. Inside the ovary, ovules are present. When the ovary fertilized produces fruits, when the ovule fertilized produces seed. The simplest definition for seed is a fertilized ovule is called as seed. And one more, the general structure of seed consists of three main parts, such as seed coat, endosperm, embryo. When the ovule or megasporangium is fertilized, it produces seed. The general structure of seed consists of three major parts, such as seed coat, endosperm, embryo. Seed coat. Outermost layer of seed is called as seed coat. When the ovule is fertilized, along with ovule, the integuments of ovules also will fertilized. Outer integument of ovule converted into outer seed coat, inner integument of ovule converted into inner seed coat. Outer seed coat is called as a testa, inner seed coat is called as tegmen. For example, the structure of seed in dicots or in dicotyledons, in general if we discuss, this is the basic structure of dicotyledon seed. Any structure of seed, either monocotyledon seed or maybe dicotyledon seeds, are having three major parts: seed coat, endosperm, embryo. The outermost structure of seed is called as seed coat. Seed coat is a protective structure. The origin of seed coat from the integuments of ovule. Region is the ovules fertilized into seed. Then integument also fertilized into seed coat. When the integuments converted into seed coat, the seed coat main function is protection. So, the outer seed coat is called as testa. The origin of testa ontogenetically from outer layer of integument of ovule. The inner layer of seed coat is called as tegmen. Ontogenetically, the origin of inner seed coat or tegmen from inner layer of ovule or inner integument. These are the origins of seed coat, testa and tegmen. The main function of seed coat is protection. And one more important characteristic feature. In the structure of ovule, two integuments are leaving a narrow space is called as micropyle. In the structure of ovule, micropyle is present. After fertilization, it's converted into seed. That micropyle it's converted into seed pore. The main function of this micropyle or seed pore, it's responsible for intake of oxygen or exchanges of gases, and it absorbs water from the soil for during seed germination. This is the function of micropyle or seed pore. Then next, the second major part of structure of seed is endosperm. In dicotyledon seed, endosperm is a nutritive tissue. It's a product of central cell in embryo sac. It's a product of triple fusion. The ploidy of this endosperm is triploid in angiosperms, haploid in gymnosperms. In the structure of seed, the endosperm is present or absent. For example, this is endosperm. In the structure of seed, the endosperm is present or absent. It's one of the nutritive tissue. It's responsible for nourishing growing embryo. These endosperm present or absent, because of this characteristic feature, the seeds is classified into three types, such as endospermic seeds or albuminous seeds, non-endospermic seeds or exalbuminous seeds, perispermic seeds. Let's we discuss the first type of seed is endospermic seeds, is also called as albuminous seeds. For example, this is an embryo. Even after maturation of embryo in a seed, still endosperm is present. Such kind of seeds are called as endospermic seeds or albuminous seeds. Example, rice seed and wheat seed and barley seed and maize seed. If in case before maturation of embryo, if endosperm is completely consumed for their growth and development of embryo, but embryo is not still, it's not mature. Before maturation of embryo, if endosperm is completely consumed, such kind of seeds are called as non-endospermic seeds. Reason is, at mature condition of seeds, there is no endosperm. Such kind of seeds we called as non-endospermic seeds or exalbuminous seeds. Example, all Fabaceae family member seeds like pea seeds, bean seeds, groundnut seeds are non-endospermic or exalbuminous seeds. The third type of seed is perispermic seeds. Example for perispermic seeds is beet and black pepper. Beet and black pepper are the best example for perispermic seeds. Sir, what is perispermic seeds, sir? Generally, in the structure of seed, the nutrition for the growing embryo is endosperm. In non-endospermic seeds, before maturation of seed, endosperm is completely consumed, but still embryo is not yet mature. In such kind of seeds, for further maturation of seeds or further maturation of embryo, further growth of embryo, nutrition is required. That nutrition coming from the nucellus. Whenever the food getting nucellus, from the nucellus, then embryo going to mature. Even after maturation of seeds or maturation of embryo, in mature seeds, still if nucellus is present, such kind of seeds are called as perispermic seeds. Once again, the simplest definition or the meaning of perispermic seeds. In non-endospermic seeds or exalbuminous seeds, before maturation of embryo, endosperm consume completely happen or endosperm is completely utilized. In such kind of seeds, for further growth of embryo, food material is required. That food material from nucellus. Then embryo going to mature. After maturation of seeds, if the seed still it has nucellus, such kind of seeds are called as perispermic seeds. Simplest definition is the remnant of nucellus in mature seeds is called as perispermic seeds. The remaining nucellus present in the mature seeds is called as perispermic seeds. The ploidy of nucellus is diploid and perisperm also diploid. The remnant of nucellus in mature seeds is called as perispermic seeds. Example, beet, black pepper. This is a basic information about endosperm. The third characteristic features for this seed is embryo. Embryo is one of the most important characteristic feature in seed. Its ploidy is diploid. It is responsible for formation of young seedlings. It's responsible for continuity of generation from one generation to another generation. If there is no embryo formation, there is no next generation. So embryo, its ploidy is diploid. It's a third structure of seed. It's responsible for continuity of generation. It's responsible for production of young seedlings like plumule and radicle. Plumule can develop into shoot system, radicle can develop into root system. For example, if you see the peripheral structure of dicotyledon seed, the structure of dicotyledon seed having two cotyledons. That's the reason this seed is called as dicotyledons. The embryo having embryonal axis, it is having plumule and radicle. Radicle develops into root system, plumule develops into shoot system. So the ovule fertilized into seed. Then the micropyle of ovule converted into seed pore. Seed pore main function is exchanges of gases. During seed germination, it absorbs water from the soil. That is the role of seed pore or micropyle. And the hilum, the junction in between stalk of the ovule and body of ovule is called as hilum. After fertilization, it forms scar of the seed. Scar of the seed. And this is another one more sectional view of dicotyledon seed. In the structure of dicotyledon seed, it has two cotyledons. And it has nutritive tissue endosperm, and it has hypocotyle, and it has root axis, and it is surrounded by a protective coat is called seed coat. Outer seed coat is called testa, inner seed coat is called tegmen. The origin of testa and tegmen ontogenetically from outer integument and inner integument of ovule. This is a basic information about seed and in dicotyledon plants. Let's we discuss an information about structure of seed in monocotyledons. In general, this is the basic structure of seed in monocotyledon plants. In monocotyledon plants, the number of cotyledons is only one. That single cotyledon is called as scutellum. Its position is lateral side. Yesterday we discussed. And one more important characteristic feature. These monocotyledon seed, outermost covering part is called as pericarp, is also called as fruit wall. During fertilization, ovary converted into fruit, ovule converted into seed, ovary wall converted into fruit wall. Fruit wall is nothing but pericarp. Its function is protection. And all monocotyledon seeds are full of endosperm. And this is an embryo. The embryo and endosperm separated by a proteinaceous layer in monocotyledon seeds is called as elluran layer. Presence of elluran layer in the structure of monocotyledon seeds, it's an important unique characteristic features of monocotyledon seed. The ploidy of endosperm is triploid. The proteinaceous layer, elluran layer also, it is immersed in endosperm, then its ploidy also triploid. And the embryonal axis having plumule and develops into shoot system, and it has a radicle develops into shoot system. In monocotyledon, the radicle surrounded by a sheath is called as coleorhiza and a plumule surrounded by a sheath is called coleoptile. Presence of coleorhiza and coleoptile is the characteristic features of monocotyledon. This is an information about structure of seeds. Now we discuss what is dormancy and what is seed germination, what is the importance of seed, what is the significance of seed, what is the viability of seed, we discuss now. What is seed dormancy? Generally, seed dormancy means a state of inactivity of embryo. For example, this is the structure of dicotyledon seed. Simple example, structure of dicotyledon seed. Outermost seed coat is called as testa. Inner seed coat is called as tegmen. Ontogenetically, the origin of testa and tegmen from the ovules in angiosperms. And it has an opening pore is called as micropyle or seed pore. Embryo has a young structure for formation of seedlings like plumule and radicle is embryo. The ploidy of this embryo is diploid. This structure generally responsible for continuity of species, that means formation of next generation or next plant. If this embryo is inactive, not activated or inactive, when the embryo is going to inactive state, then the seed is unable to germinate. Such kind of phenomenon is called as a dormancy. Dormancy means a state of inactivity of embryo, then a seed is not able to germinate. When the embryo is going to inactive state, then the seed is unable to germinate. Such kind of phenomenon is called as a dormancy. When the seed is going to dormancy, in that seeds, the moisture content, that means water content, it reaches up to 10 to 15%. When the water content is decreases 10 to 50, up to 10 to 15%, then there is no rapid metabolic activities, then the seed is going to resting stage, that is called as dormancy. Clear? Then the second concept is seed germination. What is the seed germination? For this seed, for these inactive seeds, if you are providing an optimum conditions, like optimum environmental conditions, like adequate or optimum moisture, that means water content, moisture, and optimum oxygen and suitable temperature. When there is a availability of suitable temperature, suitable oxygen and optimum moisture to this seed, the seed is going to germinate. That phenomenon is called as seed germination. Let's we discuss what are the advantages of seed to angiosperms. Generally, there are 4 basic advantages of seeds in angiosperms or in flowering plants. The first advantage is seed have better adaptive strategies for dispersal to new habitats. For example, this is one seed. This seed is formed in one area. If I bring this seed from this area to another area, if I keep here, a new plant will arise. Because of seed, a new generation is created in the new area than compared to existing area. So, for dispersal of new habitats from one area to another area because of seeds. Once again, I repeat the first advantages of seeds to angiosperms. Seeds have better adaptive strategies for dispersal to new habitats. Maybe the seeds eaten by any birds or any animals. If these animals move from one place to another place, if the seeds come out of the body of animals through the excretion, a new habitat will be started in new area. by the seed. It is the first adaptations or advantages of seeds to angiosperms. The second advantages of seed in angiosperms. Generally, seed has sufficient storage of food, that is endosperm and nucellus. Whenever seed has a sufficient food like storage of food or endosperm, this endosperm is nourishing to growing embryo. That is the second advantages of seed to angiosperm. Character is it has a sufficient food reserves for nourishment of young seedlings or young embryos or young seedlings like plumule and radicals, because of seed can store food material. The third advantages of seed is due to presence of hard seed coat like testa and tegmen. These testa and tegmen seed coats, it's giving protection to young embryo. This is the third advantages of seed. Protection is provided to young embryos by the hard seed coat like testa and tegmen. And the fourth characteristic feature or advantages of seed to angiosperms is the seeds are formed during sexual reproduction. Then meiotic cell division must be taken or must be takes. Whenever meiosis takes place during sexual reproduction, a new variations are created in the next generation. Seed is the product of sexual reproduction. Meiosis compulsory can takes. Then it creates or it generates new genetic recombinations as it is a product of sexual reproduction. These are the four advantages of seed. Once again, a brief recall, four advantages of seeds to angiosperms. First advantages is the young embryo protected by hard seed coat. That is the first advantages. Seeds can reserve food material for growing young seedlings like plumule and radical. And seeds are the having better adaptive strategies for dispersal to new habitats, other areas. And seeds are the product of sexual reproduction. During sexual reproduction, meiosis cell division can occur. Then it creates recombination. So, due to seed formation in angiosperm, it generates a new recombinations because it's a product of sexual reproduction. The last and final most characters of seed here. Seed is the basis of our agriculture. It's one of the important character. So, seed is a basis of our agriculture. Reason is seeds are showing dehydration and these are showing dormancy. Because of seeds are showing these two characters, these seeds are preserved throughout the year for our food, and these seeds are used as continuation of species from one season to another season. These are the two characters responsible for seed is a basis of our agriculture. Let's we discuss seed viability. What is seed viability? The definition for seed viability is the period for which the seeds retain their power of germination is called as seed viability. After fertilization, when ovules are fertilized into seed, after formation of seed, these seeds having the power of germination capacity. Once again, the time or period for which the seeds, how much time can retain a seeds for their germination is called as seed viability, or the period or the time in which seeds have the power of germination capacity is called as seed viability. The seed viability is not constant for all the species of plants. It varies from one plant species to another plant species. Some of the examples I would like to discuss here. For example, in the plant of oxalis, it produces seeds. After formation of seeds from the oxalis, these seeds are having the viability or power of its retaining capacity for seed germination is few months. But whereas in other plant species, for example, one more, other plant species are having a power of germination capacity, time duration, several years, maybe the recent records of very old yet viable seeds are finding. For example, here I am giving two examples, A and B. Several records of very old yet viable seeds, I am giving two examples here. The oldest viable seed is lupine. Scientific name is Lupinus arcticus. It was first identified from Arctic tundra region. From the Arctic tundra region, the oldest viable seed was identified and recorded as a viable seed. So, the lupine is the oldest viable seed. This seed germinated and flowered after an estimation of 10,000 years of dormancy. When the seed is formed from the Lupinus arcticus or lupine, up to 10,000 years, it has the retained capacity or power of germination capacity. After 10,000 years of dormancy, then it will be germinate and flowered and produces new plant. So, the viability of lupinus is, the seed viability of lupine or Lupinus arcticus is 10,000 years. It is the oldest yet viable seed in a plant kingdom. The second oldest viable seed is recorded from the place of King Herod's palace. The King Herod's palace, it is located near to Dead Sea. When the archaeological scientists, they went to Dead Sea, they visit King Herod's palace, they identified one seed. The name of seed is date palm. Scientific name of date palm is Phoenix dactylifera. The Phoenix dactylifera or date palm, it was identified recently by archaeological scientists. It has the viability is 2000 years viable. Up to 2000 years, it is going to dormancy, then after it will be germinate. That is the viability of seed in date palm. The question rising in exams is entirely different. Which of the following seed is oldest yet viable seed? Which of the following seed is recent oldest seed? The oldest yet viable seed is lupine or Lupinus arcticus. It has the power of germination capacity 10,000 years of dormancy. It was identified from Arctic tundra. Which of the following seed is the recent oldest viable seed means date palm, scientific name Phoenix dactylifera, its viability is 2000 years. It was first identified near to Dead Sea in the palace of King Herods by recent archaeological scientists. Now we discuss some of the seeds. For example, orchid fruit. Orchid fruits are having thousands of very tiny or smallest seeds. The smallest seeds we are seeing in one of the plant or one of the family Orchidaceae, example orchid. The orchid fruit has many tiny or microscopic seeds. Whereas some more parasitic plants such as Varabanca and Striga, these are the parasitic plants. These are also having the smallest seeds are tiny seeds. Now we discuss the last and final most concept in sexual reproduction in flowering plants is apomixis and polyembryo. What is apomixis? According to Greek language, the heading of apomixis having two words. One is apo, another one is mixis. In Greek language, apo means without. Mixis means union or mixing. Without union or without mixing is called as apomixis. So, what are the structures is not mixing? What is the new structure is formed in which family we discuss now? Generally, apomixis is a special mechanism. During apomixis, produces seeds without fertilization. This is the character. Generally, seeds are produced from the ovule during fertilization. If seeds are produced from the ovule without fertilization is called as apomixis. Apomixis is a special mechanism. Without fertilization, seeds are produced means there is no union of male and female gametes. Without mixing of male and female gametes to form a seed is called as apomixis. Apomixis is a special type of mechanism. It is seen in a group of family members such as Asteraceae and grasses. In the members of Asteraceae and grasses, a specialized mechanism we find, that is apomixis. During apomixis, seeds are formed without fertilization. And the next, it is a form of asexual reproduction. If there is a fusion of male and female gametes is called sexual reproduction. Without fusion of male and female gametes is called asexual reproduction. Then apomixis is not a sexual reproduction, it is a asexual reproduction. But in plants, it is mimicry or mimics to sexual reproduction. But it is a asexual reproduction, it is a mimicry to or mimics to sexual reproduction. So, during apomixis, a diploid embryo is formed in the seed. How? Because of this characteristic feature, how diploid embryos are formed without fertilization in apomictic seeds. From this character, apomixis is of two types. One is adventive embryonic or sporophytic budding. Second one is recurrent agamospermy. So, what is adventive embryonic or sporophytic budding? If you see clear, this is the structure of ovule. This is female gametophyte or embryo sac. In angiosperms, the female gametophyte or embryo sac is 7-celled and 8 nucleated. It is surrounded by integuments, outer integument and inner integuments. And one more, the embryo sac having egg cell. The ploidy of egg cell is haploid. Here, during apomixis, there is no fusion of haploid male gamete with haploid female gamete or egg cell. That means, during adventive embryonic, other than egg cell, other parts of the ovule can develop into diploid embryo. What are the other parts of the ovule? Such as nucellus, a mass of parenchymatous nutritive tissue is called as nucellus. Nucellus ploidy is diploid. And one more, ovule is a megasporangium. It is surrounded by protective layers is called as integuments. Integuments ploidy also diploid. Other than egg cell, reason is there is no fusion of egg cell and male gametes during apomixis. Other than egg cell, other parts of the ovule can develop into diploid embryo. Such kind of apomixis is called adventive embryonic or sporophytic budding. Example, citrus plant, mango plant, opuntia plant. Sir, what is adventive embryonic? Other than egg cell in a ovule, either from diploid nucellus or maybe diploid integuments can produce as a diploid embryos is called as adventive embryony. Other than egg cell, other parts of the ovule such as diploid nucellus and diploid integuments can develop into diploid embryo during apomixis is called as adventive embryony or sporophytic budding. Example, citrus, mango, opuntia. What is the second type of apomixis? During apomixis, seeds are formed without fertilization. The seeds having diploid embryo. The second method of seeds are having diploid embryo by apomixis is recurrent embryo or recurrent agamospermy. The recurrent agamospermy we are seen in apple plant and pear plant. The plant of apple and pear having the second type of apomixis is recurrent agamospermy. Sir, what is recurrent agamospermy, sir? Generally, megaspore mother cell is diploid. Clear? Without meiosis, the diploid megaspore mother cell directly develops into a diploid embryo sac. Generally, embryo sac is haploid. But in this method of apomixis, the diploid megaspore mother cell directly develops into diploid embryo sac. Then diploid embryo sac having diploid egg cell. Then the diploid egg cell without fertilization, without fusion of gametes, then it forms a diploid embryo. Then diploid embryo present in apomictic seeds from diploid egg cell from diploid embryo sac. This phenomenon is called recurrent agamospermy. Once again, I recall what is the second type of apomixis. The second type of apomixis is recurrent agamospermy. Recurrent agamospermy seen in apple plant and pear plant. Generally, in angiosperms, megaspore mother cell diploid undergoes meiosis, produces 4 haploid megaspore. Out of 4, 3 are degenerated. Only 1 haploid megaspore is functional. That develops into haploid 7-celled 8-nucleated embryo sac. Then egg cell is haploid. But in this case, the diploid megaspore mother cell directly develops into diploid embryo and it has the diploid egg cell. That diploid egg cell without fertilization develops into diploid embryo. This kind of apomixis is called recurrent agamospermy. Let's discuss what is polyembryony. Generally, poly means many. Embryony means embryos. In general, one seed has one embryo. If one seed has many embryos, is called as polyembryony. The definition is occurrence of more than one embryos in a seed is refers to as polyembryony. Present generally one seed has one embryo. If one seed has more than one embryo, is called as polyembryony. This polyembryony was first discovered by one of the dynamic scientists. His name is Leeuwenhoek in the plant of citrus. The scientist Leeuwenhoek first discovered polyembryony in the plant of citrus. During polyembryony, zygote, when fertilization can take place, haploid male gamete with haploid female gamete to form diploid zygote, the diploid zygote can develop into diploid embryo. It's a normal embryogenesis or embryo development. But along with the zygote, other diploid sporophytic tissue, nucellus can form diploid embryo. Integuments also forms a diploid embryo. So here one seed has many embryos. This phenomenon is called polyembryony. During polyembryony, except zygote, from nucellus and integuments, embryos are formed. All these embryos morphologically and genetically similar, those are called as clones. If the embryos are morphologically, genetically similar is called as clones. Except zygotic embryo, the rest of embryos formed from nucellus and integuments morphologically similar, genetically similar. This is an information about apomixis and polyembryony. At the end of sexual reproduction in flowering plants, second chapter in second year syllabus, we discuss a peripheral ploidy of different parts of the plant. For example, this is a bisexual flower. Flower is a part of sporophytic plant. Then its ploidy is diploid. And the pedicel also diploid, thalamus also diploid, calyx also diploid, corolla also diploid, stamens also diploid, pistil or carpel also diploid, anthers also diploid. Because of flower is diploid, floral parts also diploid. And in anther, there is microsporangia is present. Inside the microsporangia diploid, microspore mother cell or pollen mother cell is present. Its ploidy is diploid. Just we can recall ploidy of different parts of the concept. So the ploidy of microspore mother cell or PMC is diploid. Whenever it is undergoes meiosis, it produces 4 haploid pollen grains. Then the product of meiosis always haploid. Pollen grains or microspores is haploid. Then undergoes mitotic division to produces 2 unequal cells, vegetative cell and generative cell. The ploidy of vegetative cell or tube cell is haploid. The ploidy of generative cell nucleus also haploid. Then it is undergoes second mitotic division. Then in the pollen tube, 2 haploid male gametes are formed. Then the male gametes ploidy also haploid. And here the innermost layer of bisexual flower, gynoecium, it's diploid, and inside it has ovules. Ovules also are diploids, and integuments also are diploid, and nucellus also are diploid. Some of the nucellus cells modified into megaspore mother cell diploid. Whenever it is undergoes meiotic cell division, it produces 4 haploid megaspores. All these products of meiosis is haploid. So megaspores ploidy is haploid, megaspore mother cells is diploid. Then megaspore, haploid megaspore can develops into embryo sac. Then ploidy of embryo sac also haploid. In that, the egg cell also haploid, and synergids also haploid. Polar nucleus is haploid, 2 polar nucleus. Secondary nucleus means before fertilization, this 2 polar nucleus is fuses to form secondary nucleus. Ploidy of secondary nucleus is diploid, polar nucleus is haploid. The ploidy of antipodals is haploid. These are the characteristic features of ploidy of different parts during sexual reproduction. Thank you.